everyone, to the people here in the room, also to the people in the stream. We are back in a new edition of the Liver Seminars 2026 in the winter edition, and we have the pleasure to have here Louise Thomsen. Karen is a clinical professor in experimental hepatology in the Department of Clinical Medicine in the Aarhus University in Denmark. She completed her medical studies in 2002 in the Aarhus University. Then later she completed her PhD in 2012. And then he got some clinical training, right, to become a specialist in gastroenterology, hepatology, 2016. She also made a one-year stage in London. And I think from there, she did all her professional career in Aarhus. When it comes to research, she is very known for studying the contribution of inflammation, the pathophysiological impact of inflammation into the liver and in other organs like the brain during liver disease. She studied some mechanisms which were related to the synthesis of urea in chronic liver disease. She also described that during MASH there is a downregulation in ureagenesis, and it affects the homeostasis of nitrogen, and then it results in hyperammonemia. And more recently, she described that this hyperammonemia is correlated with cognitive dysfunction in muscles, in animal models, and also in humans. So in summary, Karen is a clinician scientist who reaches some patient-based clinical studies with mechanistic laboratory research, and her contributions in liver inflammation, in metabolic deficiency, and in cognitive dysfunction puts her in the forefront of translational hepatology research. So thank you for coming, Karen, and whenever you are comfortable, you can start.
Thank you so much for that kind invitation to come here today and a nice introduction. I'm really looking forward to participate in this year's liver seminars here in Madrid. Looking forward to the next couple of days. My talk is about cognitive dysfunction in metabolic dysfunction associated steatotic liver disease. And as we all know, MASLD is increasingly common and now actually affects more than 30% of the general population all over the world. And as we know, it's closely associated to obesity and to the metabolic syndrome. And actually now in the US, the second most common indication for a liver transplantation is now MASLD cirrhosis. So really a common disease. We see MASLD as a hepatic manifestation of the metabolic syndrome. And as you can see, it's really indeed a multi-system disease. Almost all organs are affected by obesity, by insulin resistance, and by dyslipidemia and so on. And we have to bear that in mind when we talk about cognitive dysfunction in MASLD. Is there actually an independent effect from the liver, or we will see during this talk the question to that. MASLD is a wide spectrum of disease, going from simple steatosis just with triglycerides in the hepatocytes, over inflammation and cellular damage as we see it in steatohepatitis, and a subgroup also develops fibrosis, cirrhosis, and even HCC. The prognosis of patients with steatosis seems pretty benign, whereas patients with steatohepatitis, they have an increased liver-related morbidity and mortality. And this study here is from Sweden, from Mattias Ekstedt, showing, I think they had more than 200 biopsy-proven MASLD patients, and they showed that the patients with more than stage 2 fibrosis, these patients, they have increased mortality. And that has been shown in other studies as well. So it seems that development of fibrosis is the most important contributing factor in MASH-associated mortality. We know in cirrhosis that portal hypertension is associated to mortality, but what about in non-cirrhotic MASLD? This is what this is about. This study is from Sven Franke from 2010, where they included patients with non-cirrhotic stage of MASLD, and they actually found increased portal pressure in the patients with grade 3 steatosis. So the question is, complications related to portal hypertension, is that also seen in non-cirrhotic MASLD? And what about the metabolic liver function? Is that affected in MASLD? And again, talking about non-cirrhotic MASLD. Several studies have investigated the activity of the microsomal system. This is just one of the many studies using various breast tests. This is the methastatin breast test. And in this study, they found that MASH patients, they have a reduced ability of the liver to actually convert this compound compared to controls and also compared to patients with steatosis. In Aarhus in Denmark, we have done a similar study. We used the aminopyrin breast test in a cohort of patients of MASLD, and again we found progressive reduction in the ability to convert this compound in the liver, lowest in MASH patients, but also reduced in patients with simple steatosis. So there seemed to be a problem with the microsomal system. I have found this old study from the 80s where they looked at another liver function, that's the galactose elimination capacity, which takes place in the cytosol. And in this study they found that patients with steatosis, they actually also have a reduction in galactose elimination capacity compared to controls. But you can see there is a further reduction in cirrhosis patients. So to further investigate this, we performed some animal experiments. And first a bit about this animal model, which we did maybe 10 years ago, where we induced in V-star female rats, we induced steatohepatitis by feeding the rats a diet with a high concentration of fat and cholesterol. And we really like this model because it's a Western style diet. It's a Western style way of inducing MASL. No artificial deficiencies of any nutrients are used, and no noxious toxins, which also sometimes actually affect the brain. So this is a good model to use. And just a bit of data from that model, you can see that the liver size is clearly increased also already after 4 weeks. And also the liver is full of fat. We actually see this almost after 1 week when we did the short model. And when you look at the liver weight, that had doubled in size after 4 weeks, and after 16 weeks, 3 times the size of the controls. So the fat really goes to the liver. And in this model, we also found an increased spleen weight, maybe a sign of portal hypertension in this model. Unfortunately, we didn't measure the portal pressure here. And this is the histology. We see that the diet induces steatosis and inflammation, and after 16 weeks also bridging fibrosis. So it is a model with a histology similar to human MASL. Then we looked more into metabolic liver function, and this is the essential function called urea synthesis. And we measured in the liver the 5 different enzymes catalyzing urea synthesis shown here, and they were all reduced at the gene level in liver tissue. We also measured 3 of them, the most important ones, at the protein level, and found that CPS and OTC were both reduced in this model. These 2 enzymes are both located in the mitochondria. And then we did some in vivo measurements of the capacity of urea synthesis. And we do that in the rats by infusing alanine, and then we remove the kidneys before we start to avoid excretion of the urea into the urine. And again here we found a reduction of urea synthesis, and that might impair the rats or the human beings' ability to adjust to changes in requirements to the nitrogen homeostasis. For example, during stress or during infections or changes in diets. But unfortunately, we didn't measure plasma ammonia in this study. Just a bit about urea synthesis here. It's an essential liver function that takes place both in the mitochondria and the cytosol, and it has a central role in amino acid metabolism, and amino nitrogen not used for protein buildup is eliminated via the synthesis of urea. And it is an irreversible process, as human cells, they cannot break down urea, so it is the final step in the elimination of nitrogen. Urea is then excreted into the urine. And we have performed studies. This is an old study from Hendrik Wilstrup, my professor, from the 80s, where he showed that the urea synthesis is reduced in patients with cirrhosis. You can see that the urea nitrogen synthesis is reduced in relation to the amino nitrogen concentration in cirrhosis patients, which are the lines, and that hatched area there, that's the controls. And that's well known in clinical hepatology, because that's one of the reasons why patients with cirrhosis, they get hepatic encephalopathy due to this reduced ability to produce urea. And we have later done another study in patients with alcoholic hepatitis, and we found a severe reduction in urea synthesis in these patients. Here we measured a thing called functional hepatic nitrogen clearance, where we again infuse alanine in the patients. And here we don't remove the kidneys, but we just collect all the urine being produced during the infusion and measure urea in the urine to correct for that. And this reduction was actually similar to patients with acute liver failure, so a severe reduction in these patients. Our findings in the rat model were confirmed in another model. This is in collaboration with Rajiv Jalan's group in London, where they did a study in mice, the MCD mice. And they also in this model found a reduction in one of the urea cycle enzymes, the one we measured, OTC. And in this study, they fed the mice with carbon to try to reduce bacterial translocation. And as you can see, the carbon treatment restored OTC gene expression in these mice. Then we looked at the time course of changes in the model. As you see below, that's the histology from these rat experiments, again performed in London with Rajiv Jalan. And here you can see, if you notice, the fibrosis in these rats are actually much more pronounced than the ones I showed first. These are Sprague-Dawley male rats, and maybe that's why they become more fibrotic. And again, we measured urea synthesis, here the OTC activity, and found a reduction in the activity of this enzyme. And this time we also measured ammonia, both in the plasma and in liver tissue using Nessler staining. And as you can see, plasma ammonia and ammonia in liver tissue is actually increase here in this model. Then we did more studies looking at the potential reversal of the urea cycle changes. We fed this time the rats high-fat, high-cholesterol diet for up to 12 months. And again, we found a reduction in OTC and CPS after 10 and 12 months at the gene expression level and also at the protein level. And we again measured OTC enzyme activity and found similar findings. And this time we then switched back the diet after 10 months, back to normal diet, and saw that the histology became much better. They recovered. And also the changes in urea synthesis here was also restored after reversing the diet. So the changes are reversible, at least in rats. Then we moved into human beings. These are from patients with liver steatosis and MASH. And again, we did OTC staining with immunohistochemistry and also measured the OTC enzyme activity and found again that that was reduced both in patients with steatosis and in NASH patients. And in Aarhus in Denmark, we did the same in another cohort of MASH patients. We did RNA-seq here and measured not just the 5 enzymes involved in urea synthesis, but also more enzymes involved in amino acid metabolism. And again, we found reduction in simple steatosis and in the MASH patients. And this time we also found an upregulated glutamine synthase, probably as a compensatory mechanisms. Again, we performed the in vivo capacity for urea synthesis, infused alanine into the patients, collected the urine, measured urea, and again found that the urea synthesis was reduced. And in another cohort of patients, not this cohort because we didn't measure ammonia there, we found increased ammonia levels both in patients with steatosis and in patients with NASH compared to controls. And that was both in plasma and also below in liver tissue. So these changes in urea synthesis and changes in ammonia levels, could that actually lead to cognitive changes in MASLD patients? This is the first study pointing to this. This is from Filippo's group in Valencia, an older study. And there he found that in NASH patients or MASH patients, they had increased ammonia, as you can see, compared to controls and patients with steatosis. And in half of the MASH patients, they also had cognitive changes. He used the PSE test. It was only a small cohort, but half of them actually had some cognitive changes. The first studies looking more into potential difficulties with cognition in a larger group of MASLD patients were from Julia Newton in Newcastle. She did lots of studies, amongst others this, where they found that MASLD patients, they have severe problems with fatigue and daytime somnolence. She measured the fatigue impact scale, which scores 40 different daily activities and how affected they are due to fatigue. And as you can see, that score is increased in MASLD patients compared to controls. And a follow-up study a few years later, she found that almost half of the MASLD patients had a score above 40, which is considered significant fatigue. And you can see she included other chronic diseases with severe fatigue, PBC, and chronic renal failure, which are well known for fatigue, and found changes to the same level as in MASLD patients. In another study from the same group, they found functional impairment in 70% of MASLD patients. And that was again difficulties with activities of daily living. And this time they measured the PAC score, which is the physical ability to perform 20 daily activities divided into 8 validated domains as shown here. And you can see that difficulties were just as pronounced as in another cohort of patients in alcoholic liver-related disease. And if you notice over here, 8% of the patients in the MASLD cohort had cirrhosis, and that was the case in 65% of the alcoholic liver disease cohort. So actually, without having cirrhosis, they have functional impairment, these patients. Many smaller studies have used different general dementia screening tools like minimal mental state examination and the MOCA test. And most of these studies, they have found a global reduction of cognition in these patients, but most of the studies are way too small to adjust for potential confounding. Since then, many more some population-based studies have looked at the association between MASLD and dementia, and some find an association, especially in the younger age group, and others no association after adjusting for confounders. So it's not quite clear if there is an association here. Focusing more specifically on cognition, this is the first study to be performed from the US, including more than 5,000 participants, again in a population-based study. And they excluded patients without an ultrasound on the liver, and patients with a high alcohol consumption, and patients with Hep B and C. And then they found more than 4,000 participants where 20% had MASLD. And then they performed 3 different computerized cognitive tests, and they found that the MASLD patients had a lower performance in the serial digit learning test. And that was independent of cardiovascular disease and its metabolic risk factors. Then they excluded the patients with an intermediate and a high risk of fibrosis, and they actually found the same results in the patients with a low risk. And that was evaluated from the NAFLD fibrosis score, which is blood samples and other parameters. Another study looking at cognition is a study from Denmark, from Mette Munk Lauritsen's group, where they included 180 patients in a bariatric study. They performed liver biopsies of all of them, and almost 80% had MASLD and 2% with cirrhosis. And then they performed the CRT, that's the continuous reaction time. We use that a lot in Denmark, also in Germany. The PSE test and a Stroop test. And in this study they found that only 8% had cognitive impairment, defined by 2 out of 3 of these tests being abnormal. But then in a subgroup of patients, they did a much broader battery of cognitive testing. That was the R-bands, and then they found that more than 40% had cognitive impairment, especially within executive and short-term memory functions. But they found no association with MASLD or MASLD severity, no association with BMI, and no association with metabolic comorbidities. Maybe this study was too small to catch that, but that was a bit surprising. And the last study I would like to mention about this is from Spain, from Valencia, where they investigated 59 biopsy-proven non-cirrhotic MASLD patients really thoroughly with a huge battery of psychometric tests. And here they found, some of them are shown here, 32% actually had mild cognitive impairment. But looking only at the PSE test, only 12% had changes. So this might point to the fact that other tests are needed when we talk about cognitive dysfunction in MASLD patients. We can't just use the same tests as we use for cirrhotic patients and minimal HE. That was impairment of attention, mental concentration, psychomotor speed, cognitive flexibility, inhibitory mental control, and working memory. Some of the same changes as we see in cirrhosis. But again, no difference between patients with simple steatosis and MASH, but this time a higher prevalence in patients with diabetes and metabolic syndrome. So I have focused a lot about urea synthesis and ammonia, but of course many other potential mechanisms are possible. This is a really nice illustration of the gut, liver, brain axis, and you can see the liver is there, importantly the adipose tissue, but also the adrenal gland, and importantly the gut with the microbes metabolites, and the enteric nervous system. But the question again is there actually an independent effect of the changes in the liver affecting cognition in MASLD patients? A potential mechanism is gut dysbiosis leading to systemic inflammation. And as we know, the gut microbiome in MASLD patients is altered with a reduced bacterial diversity and also a loss of so-called beneficial microbes in the gut, and at the same time an increase in pathogenic bacteria. And that's the same thing we see in liver cirrhosis, maybe more pronounced in liver cirrhosis. And this cartoon here shows the link between dysbiosis and inflammation. And as we know, the liver and the gut is really intimately linked due to the fact that the liver receives the majority of its blood via the portal vein going to the liver. And you can see here that dysbiosis can affect the intestinal permeability, increase the permeability, leading to bacterial translocation and endotoxins and other bacterial products coming into the circulation. It also changes the short fatty acids and the composition of the bile acids, and all this can lead to systemic inflammation and also to development of steatosis and steatohepatitis. But it's not only the gut contributing to the systemic inflammation. Of course, also the inflamed liver itself in muscle contributes, and the inflammation in the adipose tissue due to insulin resistance. And then the question is, can systemic inflammation actually lead to affected cognition? Can it lead to neuroinflammation? And it has been shown that increased free fatty acids in MASLD and systemic inflammation actually increases the blood-brain barrier permeability, and in that way can activate microglia cells. Microglia are the macrophages in the brain. And that way also affect neurons and their cell signaling, and maybe also lead to neurodegeneration and brain atrophy, as shown in other brain diseases like Parkinson's and Alzheimer's disease. Several studies have actually looked at the association between systemic inflammation and cognition. This is again a population-based study from Korea. More than 4,000 participants, 30% had MASLD, this time evaluated from the fatty liver index. They performed the minimal mental state examination test, and they found an association between cognitive impairment and inflammation. And that association was strongest in the patients with the highest inflammatory status, as you see there, with the highest white blood counts and the highest CRP. This is another study. It's actually the same cohort from Valencia we saw before, with the 59 MASL patients. They looked after potential mechanisms for the mild cognitive impairment they found in 30%. And they looked at the immunophenotype and cytokines and so on. And they found increased activation of the CD4 positive lymphocytes, and mainly the Th17 subtype, and also increased plasma levels of pro-inflammatory cytokines, especially IL-17 and IL-21, IL-23, and also some anti-inflammatory cytokines, IL-10, and also a higher expression of the chemokine receptor CCR2. And it is known that plasma IL-17 can increase the permeability of the blood-brain barrier and thereby allow for infiltration of immune cells, and also activate microglia, astrocytes, and cause neuroinflammation. And maybe also the increased CCR2 and CCL2 may promote monocyte infiltration into the CNS, but these are just speculations, suggestions. It hasn't been shown in patients. And therefore we of course need more experiments, and we need animal experiments when we talk about the brain. And we have, in the rat model I mentioned before, performed behavioral studies in these rats. This is the forced swim test, where we insert the rats down to a cylinder full of water. First they have a test swim, I think for 15 minutes, taken up again, and then they come back into the cylinder and swim, or they are in the water for 5 minutes, and then we score for how long they climb, how long they swim, and how long they're just floating. And we found that the MASLD rats, they were swimming less and floating more compared to the control rats, as a sign of depression-like behavior. We also did the novel object recognition tests, where a rat is put into an arena with 2 different objects. It's allowed to investigate the objects. Then you take out the rat and you replace one of the objects with another object and reintroduce the rat to the arena. And then you measure how long time the rat spends investigating the new object. And we found that the MASLD rats, they spent approximately the same time investigating the new object compared to the old object. And that is, or should be, a sign of impaired memory in this model. We also looked at systemic inflammation in the same rats, and we found several increased pro-inflammatory cytokines in plasma. And we also investigated the brain using immunohistochemistry and autoradiography. And we stained in the immunohistochemistry with IBA1 staining activated microglia. And we used in the autoradiography a tracer called PK11195 that binds to activated microglia. And here we found that these microglia were activated in the prefrontal cortex in this animal model. Other groups, this is data from Anna Hadihambi in London. She has done a mouse study, a bit the same in a muscle model, where she put the mice into an arena, and she found increased anxiety, and that was shown by the fact that the mice spent less time in the center of this arena. And she also did the forced swim test and found the same as we did, that the mice spent more time floating compared to the controls, as a sign of depression-like behavior. Then she did some really nice immunofluorescence microscopy here, and she used GFAP to stain for astrocytes, and again IBA1 to stain for microglia, and found activation in both these cell types. And recently we have in our group used a new mouse model, and we did laser speckle contrast imaging, and we found that these mice, they actually have reduced brain perfusion. And we also, in collaboration with Anna Hadihambi in London, investigate our rat model, where we looked at the partial pressure of oxygen in the cerebral cortex of these rats. And we again found a reduction in the MASLD model, and that reduction actually was correlated with the results from the novel object recognition test. So it seems that these cognitive changes might be caused by reduced cerebral perfusion or reduced oxygen availability in the brain. Moving into human beings here, this again a study from Valencia, and they looked at deceased MASLD patients. And even in the early stage of disease, it's called steatohepatitis 1 to 3 in that figure, and in the pre-cirrhotic stage they actually found increased activation of astrocytes and microglia, and infiltration of both CD4 positive lymphocytes and macrophages, and a reduction in the number of neurons in these patients. And that was found both in the cerebellum and hippocampus in the pre-cirrhotic stage. And we have just done a pilot study in alive human MASLD patients using a PET MRI. In the PET scan we use that PK11195 tracer that binds to the TSPO protein. And that's a protein located in the outer membrane of mitochondria. And when microglia are activated, they upregulate this protein. And therefore it's a smart way to test for activated microglia. And it's used in other diseases like Alzheimer's and Parkinson's disease, this kind of PET imaging. And then we also did arterial spin labeling MRI on these patients to look for the cerebral flow. But only 6 MASH patients and 6 controls so far. And the MASH patients had different degrees of disease. And we can see that they have an increased hepatic fat fraction measured by MRI PDFF. And these are some of the characteristics. Of course, the BMI is increased compared to controls, and also the ALTs. And of note, also ammonia is actually higher than in the control group, but of course still within normal limits of normal. But there is a difference in the level. And these are the PET scan results using that tracer, and we can see that microglia seems to be activated in several brain regions, as shown here. And also looking at the ASL MRI, looking for cerebral blood flow, we found a reduction in that flow in MASLD patients. But these are just preliminary data, and we need some more patients included. And also we did cognitive testing in these patients as well, and we haven't looked at them yet. We have done some studies using different interventions. This is in the rat model, same rat model as described before and talked about a lot. Same changes in the swim test, the cognitive changes. And we also measured inflammation in the plasma, again found an increase in pro-inflammatory cytokines here. And this time we also measured plasma in this model, and we actually did find increased ammonia levels, and also a tendency for increased ammonia in the CSF, the cerebral spinal fluid. And then we treated these rats with semaglutide for 10 weeks, GLP-1 agonists, and saw that all these changes were reduced after 10 weeks of treatment. Then in a human study, we looked at the effects of bariatric surgery. We investigated the patients before and 18 months after surgery. We found they lost weight, all the patients, and they also were cured, you can say, from their MASLD. They all had no steatosis after 18 months. Of no, one patient actually didn't have MASLD before bariatric surgery. And then we did the cognitive testing using the PSE test. And as you can see, that's the test we usually use in cirrhotic patients. Only 4 patients actually had an abnormal test below -4 before surgery, but most of them seemed to improve their cognitive function after surgery. Then, of course, we measured urea synthesis, as talked about before. We found a reduction in urea synthesis before bariatric surgery, and that was restored 18 months after surgery. And of note, that patient up there is actually the one without MASLD to start with. Then we measured ammonia in this cohort as well, and found a reduction after surgery. As you can see, again, ammonia levels within normal limits, but they are reducing or going down after surgery. And then in another cohort of bariatric patients from another center in Denmark, we also measured ammonia and found exactly the same. So we speculate if even it's not high ammonia levels, but maybe there's a synergistic effect together with the systemic inflammation that maybe can affect the brain. And then I found this brand new study from MAPI's group in Valencia, where they also looked at cognition in bariatric surgery patients. They only investigated them after 3 and 6 months. And you can see in the PSE test, there are no changes, but using the animal naming test in the middle, B, they found a reduced percentage of patients with an altered animal naming test compared with the control group 3 and 6 months after bariatric surgery. And there were also some of the other tests that changed after 6 months. I wonder if it's a bit too early to measure already after 6 months. Lots is happening with bariatric patients when they lose weight, and these are still losing weight. So I would have advised her to wait maybe 12 or 18 months. It's difficult not to mention arteriosclerosis and cerebrovascular dysfunction when you talk about MASLD and cognitive function, because MASLD is of course closely associated with vascular complications as they share metabolic risk factors. But it has been shown that MASLD is an independent risk factor, or at least seems to be, for cardio and cerebrovascular disease, and therefore also for the vascular cognitive aging, as illustrated here. It's illustrated the effect from the liver, for example, microvascular endothelial dysfunction, inflammation, oxidative stress, and so on. Then some studies have been performed using MRI of the brain volume, and this is a meta-analysis of 4 of these studies, from the Framingham study and from the Rotterdam study. And in this study, they found an association between MASLD and the total brain volume, and that was independently associated with MASLD after adjustment for metabolic risk factors. The last study I would like to mention is this study from Sanyal, where he looked at all stages of MASLD disease. And of note, interestingly, both in cirrhosis but also in F3 fibrosis, they found that the most common decompensating event in these patients were hepatic encephalopathy. And that's interesting because usually it's ascites that is most common in other etiologies of cirrhosis. So again, maybe the comorbidities in the metabolic syndrome makes the brain more prone to developing encephalopathy. Who knows, but interesting study. So to conclude with this mechanistic drawing, I think it's shown that pre-cirrhotic MASLD is associated with cognitive dysfunction, but is MASLD, the liver affection, actually in itself an independent risk factor? I think systemic inflammation leading to neuroinflammation means something. I think that could matter, and also maybe reduced urea synthesis and increased ammonia levels. And then there's much more to be investigated regarding the microbiome and the microbiome metabolites. And then I think it's important also to maybe better map the cognitive deficits in this group of patients. So it's not just the same tests as we used for cirrhosis patients, we use for these patients. And of course, more mechanistic studies and more interventional studies are needed. And just to end with this photo, this is what it looked like when I left Denmark yesterday, -3 degrees and snow. This is the hospital, the Aarhus University Hospital. And then I just included a summer photo as well from Aarhus University to make it a bit more nice. Thank you. In MASLD patients, we don't think of that, but in cirrhosis, we, for example, treat the patients with lactulose because we know that decreases ammonia. And when patients come in with hepatic encephalopathy, that's one of the most important things to make the gut go to empty your bowels. So we use lactulose. I wouldn't advise that in this group of patients. And there are different treatments that could be used, but I think I'm not that convinced that ammonia really matters here to start to use an intervention to reduce ammonia. I don't think so. Okay. But I think, for example, semaglutide, it could be interesting to see if that makes a difference. We saw it in the rats, and I would very much like to make a human study. And these days, that's easy, right? Because it's used for soon also in Europe, I guess, on the MASLD indication to see if it actually changes the cognitive dysfunction. It's still a matter of debate if semaglutide, I don't think it can actually cross the blood-brain barrier. So the effects should come from reduction of systemic inflammation, because that's how semaglutide also works. So I think treating it should be treating the MASLD disease, and then it will follow that the cognition becomes better. Yeah, I think you're right. It's almost an impossible task actually to discriminate between that. In the preliminary pilot study we're doing, we didn't include diabetic patients to try to avoid that. So at least looking at non-diabetics, but of course overweight and inflammation in the adipose tissue and so on, I don't think we can ever come to a clear conclusion. So that's why I'm a bit reluctant to say that there really is a effect from the liver in itself. So I think it's synergistic, different things, and the most important thing is to treat the MASLD. Yeah. Results from is actually, I think they have F1 fibrosis, so it's really mild. We hoped for these rats to maybe develop F2, F3, but when we then took them down after doing all the work, we saw that most of them, they actually only have F1 fibrosis, and that was a bit disappointing. And yet then you see that even in early disease, they actually have these changes. So I think the fat really matters as well. And as you know from animal research, it's sometimes difficult to predict what you end up with when you do animal studies. And I don't know if you noticed, but the rats we did in London, they had much more fibrosis. They were Sprachdolia, not Vistar, and they were male and not female, but just also the environment or the time of the year or some noise in the animal house makes a difference. So it's really difficult to repeat the same study. But this was in very early disease, we saw this. They don't have ethanol consumption at all because we focused on that when we performed the study. But that is a recent study, right? And just even 3-4 years ago, we were not as focused on that as we are nowadays because of MET-ALD. So I'm sure many of the older studies, that's a great mix. I'm sure it is. And of course, alcohol also makes other problems in the brain. We know the patients have brain atrophy due to alcohol consumption. So it's difficult to say. And also the animal models. Now I talked about the toxins you can give. Many toxins, they actually also affect the brain. I don't know, the cholesterol we give, it's a high amount of cholesterol to actually induce fibrosis. Could that actually also affect the brain directly? So what is what? It's not easy. I can imagine after bariatric surgery where people lose weight, there must be some studies on that. Maybe Penny will tell us tomorrow. We'll see. I can imagine, but I can't just mention a study where it's actually looked at, and especially I showed some studies where they specifically looked at cognitive dysfunction as we do, but just losing weight and being overweight and move more around and stuff, there's a lot interfering, and I'm sure they improve, but is it only because of the brain getting better? Or of course we didn't perform the studies overnight, and you're absolutely right, the rats are more active, as you know from your own studies, but that's my answer is often that's why you always include controls, but because you do the same with the controls, right? So there is a difference between the two models, the controls and the muscle. But you're right, it's not easy. Yeah, we actually haven't looked at the association between the exact ammonia level and the cognitive changes and the urea synthesis capacity. I don't think we have looked into that yet. But I think ammonia, you know how difficult ammonia is to measure as well. I don't know if you noticed, but the levels measured in Aarhus in my group were actually lower than all the measures from that other cohort from Denmark. I think almost plus 15 difference in the mean. So it's really important to measure ammonia in a standardized way, in EDTA plasma, cold, measure it immediately, and so on. And that's the problem when you compare studies. Some of the other data were from Valencia, and the memory was around 100. And that usually you don't measure that high numbers. But the correlation to the urea synthesis, we should look into that. That's interesting to have a look. Yeah. And you know, even cirrhotic patients with HE, they don't develop brain edema either. If they're acute liver failure, you can of course see it. But I think the changes in a normal MRI scan would be way too subtle. But the PET scan is smarter in this connection.
Okay.
Yeah.
Okay, thank you.
We never test the patients. I only test patients in relation to our research studies. But you're right. I think doctors from these studies should focus more on that. But the test to be used, we haven't found yet, because the PSE test or the continuous reaction test or other tests are not good enough. It's not mapping the deficits these patients have. So we still have to look for which test to use and then implement it. And you're right, I think the treatment will be exactly the same as we advise the patients. They have to lose weight, they have to have their blood pressure treated and their diabetes treated and their cholesterol levels. So the treatment will probably be the same, but maybe it's easier to convince them to do something about the treatment, to also treat their brain changes. Yeah.
It has been a pleasure.